All right, good morning, everyone. My introduction is going to be quite short and sweet. But as you've probably seen on the program, we are going to we're going to hear from two different working groups that the steering committee established to try and improve our biomarker work and also help NAC with collecting biomarker data, which I think is going to be key. So the first working group you're going to hear from is the Best Practices Working Group. This is co-led by Timothy Van Meter, who is the Chief Scientific Officer at BrainBox Solutions, and Thomas Kakari, who is the Biomarker Core Director for Pittsburgh ADRC. And the charge that we gave the Best Practices Group was to update the 2014 NIA guidelines and develop updated best practice guidelines for collecting, preparing, and storing blood and CSF, and also creating a comprehensive guide to ensure that standardized assay procedures for AD biomarkers are assured so that we can enhance the reliability and reproducibility of that data. So they're going to talk about the work they've done to date, and then in our discussion we hope to get everybody's feedback so that we can wrap this working group up. The second working group that the steering committee established is data variables and data quality. This is co-led by Dr. Fanny Elahi, who is the co-director of Mount Sinai ADRC Biomarker Core, and also Dr. Kristen Russ, who is the laboratory director of the NICRAD Biomarker Assay Lab. And so the deliverables for them was to determine what fluid biomarkers are measured at each ADRC, review survey results and current materials, determine if they need to do further surveys, and I think some of you have received those surveys already, so thank you to those biomarker core leaders that have already returned those. And then determine what fluid biomarker field should be included in NAC. So what field should we be collecting in NAC that could be shareable? And so try to identify a standard set of biomarker variables, and then we're working very closely with NAC to determine the feasibility and try to work out that portal that people would then enter their biomarker data into. So you will hear their progress, and we'll have a discussion about that. And then I want to thank, we have two departing members from the steering committee, so these have both contributed greatly to the steering committee, but Jeff Daig of IU ADRC and Ricardo Osorio of NYU ADRC. They're both leaving the steering committee, probably breathing a bit of a sigh of relief that they can get that meeting off their calendar. But I really want to thank them for their hard work. With this being a new steering committee, being a new steering committee, we had to start from scratch. It was a lot of work, and so they really helped with that. In their place, I want to welcome the two new steering committee members. So Rob Rissman, who leads the USC ADRC Biomarker Core, and Tom Kakari from Pittsburgh ADRC, who leads their Biomarker Core. So welcome to them. And then for all their hard work, I think I might have gone one past. I want to thank, okay. I just want to thank the other members of the steering committee. So Dr. Lynn Beckeris of Cleveland Clinic ADRC, Carlos Cruchaga of WashU ADRC, and Tom Wingo of Emory ADRC. These are the other members. Again, thank them for all their hard work this year. And with that, we'll move into our first session. So it's my pleasure to welcome online Dr. Van Meter and Dr. Kakari, who are going to give updates on the Best Practices Working Group.
Good morning. Pleasure being with you. Hope you all are having a wonderful stay in Austin. So myself, Tim Van Meter, and Tom Kakari are going to lead you through what we've found to date following the charge that the committee gave us, as Donna had mentioned. So the Biofluid Biomarker Best Practices Work Group basically had these two goals, as Donna mentioned, to develop updated best practice guidelines for the collection, preparing, and storing of blood and CSF. There were some other biofluids that were considered, but for this effort, it was decided that we'll focus on blood and CSF, as those are the two most highly studied biofluids. And then the second goal was to create an updated standardized guideline for assay procedures, so sample handling and so forth, so that we have the proper QC methods and procedures in process, so that the results that come out of all this hard work will be comparable across centers, that investigators that are pooling results will be confident that they've been derived in a robust way, and just to have clarity on performance of how these assays are being run on the different collected biofluids. And all of this, of course, will contribute to global progress, as you've all taken part of. So what we first did was to try to assess sort of the current state and needs within the community. So in March, we put together a survey and sent it out to the 37 centers. And pretty quickly, in a couple of weeks, we got 31 different centers responding. Part of that questionnaire included a request for SOPs if people wanted to submit them. And 14 of the centers submitted SOPs from their centers that had blood and plasma SOPs, but an additional 8 centers also included CSF specific SOPs. So that was well appreciated, and we're going through those now. Another part of the questionnaire was to determine the utilization of different types of instrumentation. So I think those fell into 3 main categories, being instruments and assays that are used for discovery and characterization of disease states, high-plex sort of studies that can be found in instruments like SomaLogic and now NULISA's gaining in importance and interest, and also validation instruments such as MSD, gold standards like IPMS that help to validate the results of the original discovery experiments and look at that across large cohorts. And then there were some representation of clinical instruments like the Roche Cobas, for example. So these are the graphical results, and not surprisingly, you can see that Quanterix is one of the most highly utilized instruments across the centers. And LumiPulse being also embraced, and various other instruments also used by multiple centers. In terms of the assays being utilized, the centers reported back a lot of ATN assays as expected. Those were the most highly utilized. And we split the questionnaire into asking whether assays were run internally or whether they were sent out for analysis. Just to get a better idea of what was being done where. So bolded there are the most highly utilized assays for research at the different ADRCs. And they represent some expansion of the novel tau targets, as you've all seen in the literature. And then some growing interest in both synaptic markers and neuroinflammatory markers used at the different centers. Interestingly, what we did not see was vascular biomarkers being reported back, which of course is a really important aspect of these disease states. So with that being said, we've been collecting information. We're now formulating our sort of draft guidelines that are going to update the 2014 guidelines and being presented for review. And I'll turn it over to Tom now, who will talk about the goals that have come out of this initial analysis.
Thank you, Tim. So yeah, so as one of the key things I would like to do is looking at what are the novel markers, and then how can we try to provide a standardization. I think a goal would be that across the ADRCs, to ensure that the data sets will be very comparable. Could we have a way of standardizing how the analysis is done, and also probably how the data also would look in terms of like the values you would get out of them, and like the concentrations, for example. So the starting point for this has been to look at what is being done at this point. what sort of standardized measures are being done at the different centers. So one of the things to look at, so that the novel markers, we know that phosphatol 217, for example, is one of the key markers that people are very much interested in. Almost like all the centers got data on that. And we know it takes also a while to get that standardization all happening. Some of the novel markers, mostly tau markers, brain-derived tau, MTBR tau, and some other fragments, N-terminal and mid-region tau fragments, for example, and other phosphosites for tau, all are getting data that we would like to see how we can look at the standardization aspects. And then the increase in efforts on synaptic markers and vascular markers, also some of the sites are getting data for, and neuroinflammatory markers as well. Now, we also appreciate that it takes a while to be able to really look at how can we actually standardize the data you get from multiple instruments, and to be able to see how we can really find a way of making them quite comparable. A starting point will be to get round-robin studies, which some of the sites have started doing, and also do more cross-assay comparisons. But we believe still at this point, it's quite difficult to really get, for example, let's say if you get a value of X picogram per mil on platform A, how would it compare with Y picogram per mil on platform B, for example? So some of the criteria that we've been discussing is, one, which markers that we should choose moving forward. We should look at the weight of evidence, what sort of large cohort studies that have been done. Is it possible to look at data from multiple platforms? Is it from different centers? And if there's a commercial assay, how widely adopted has it become? And another important aspect, for example, to be able to make sense of data from different platforms and from different centers will be to get the QC data, which is extremely crucial. And we get that not all the sites use their own QCs aside from what the commercial manufacturers provide. And we're looking at ways that we could really find a meeting point to make this quite feasible, to make sure that we can get data, quality control data that will make things much more meaningful. And then the clinical ranges as well. So if you get what is like the range of the assay, and then if you get value of X versus Y, what does it mean on assay A? What does it also mean on assay B? And how accepted these assays are at the different ADRCs and also different sites. So one of the points we're looking at is like, biobanking, which is like extremely crucial in terms of what we get, because it's more like, whatever you get in, it really affects your output. So we really broken things into three major groups, say like, how do you get a specimen in the processing, and then the integrity of it in terms of the storage, and then what's the free thaw cycling. So all that happened before the sample is analyzed using whatever instrument or procedure that you would like to. And taking the sample processing or specimen processing, we have to look at centrifugation. So even from the starting point, when the blood sample is taken, how long does it take before it's processed? Is it an hour? Is it 2 hours? Is it 30 minutes? And what tubes were even used, and what processing steps, what centrifugation, what sort of centrifuge type was used, to try to be able to get consistent processing data, and to also be able to understand what are the key factors that could lead to variability in data. If one site is using slightly different approach from another site, could that be a cause for data variability? And also in terms of overnight fasting, we know that how difficult this is. Almost all the sites tell people to fast overnight before coming in, but at the same time, it's not something that you could actually police. So it makes it quite difficult. We've seen that for some of the published data show that some of the assays don't seem very, very dependent on fasting status. So whether you fast or not doesn't seem to affect it. However, we would appreciate really making sure we get to a point to say this is quite reflecting the real world, right? And also like what major changes that need to come in. Storage aspects, what freezer space is used, splitting samples across multiple freezers to prevent loss, what sort of minimal freeze-thaw cycling should we go for, and all these aspects. And how many freeze-thaw cycling should we also get? So we're trying to collect all these data from the site, and also speaking with a few key opinion leaders, and to be able to get to the point of formulating all this into one data set. So the next steps will be, to collect feedback on the initial plan that we're presenting today, and then to be able to formulate the other recommendations to be presented at AIC a few months from now, and to be able to update the 2014 guideline, which is something we had 10 years ago, and get a working document, a draft that will be put online and people can provide comments about, and we will formulate all that, we'll address all these comments to get a final product. And when that is done, another thing we consider is that whether it would also become even relevant to, in addition to putting on the NAC website, to be able to publish that as a journal article so that it makes it even much more easily citable. So yeah, we'd like to thank all the members of the work group and also the NIA reps and the NAC reps, and thanks to the Biomarker Cost-Sharing Committee of the ADRCs. Thank you.
Okay, thank you so much, Tim and Tom, for that presentation. Next is Dr. Elahi and Dr. Russ, who are going to talk about the Data Quality and Data Variables Working Group. Dr. Elahi is online, so I think Kristen has taken the lead on the presentation, and then Fanny will join us for the Q&A. Thank you, Kristen.
A little too short for this. All right, well, it's a pleasure to be here today to talk to all of you about the committee that we've been working on. So I'm a part of the ADRC Biomarker Data Variables and Data Quality Work Group, and that was co-led by myself and Fanny. Now, as we utilize more biospecimens and biomarkers in the ADRD space, it's essential to be able to share existing data. There's a lot of data, and it'll be very useful to a variety of investigators. Now, it makes sense to store the fluid-based biomarker data alongside the existing clinical data in the NAC database. But in order to do that, we're going to have to adapt and develop new data fields. So our work group was tasked with developing what those new data fields might be and how we would add them to the NAC forms to be able to utilize the data that is available across the ADRCs. In order to do this, we required data from the ADRCs. So there was an original survey sent out in 2023 that had some existing data that we were able to look at in order to help produce what variables would be utilized, and sort of get an idea of what's important to the ADRCs and how we can streamline this process. So the original survey that was sent out in 2023, 36 ADRCs responded. It provided us with information like the different specimen types that were collected across the ADRCs and what biomarker analyses were done at various labs. So you can see in the one graph up there that because we are interested specifically in plasma, serum, and CSF, I've outlined how those are utilized at different ADRCs. So you can see how many of each sample type, or how many centers of each have each sample type, that are utilizing labs within their own ADRCs, that are utilizing labs outside their ADRCs, utilizing NICRAD, et cetera. And then we found that most centers are already sharing data in some way, which is great. So we wanted to find out a little bit more about how we can make this an easier process to share with NAC as well. So what we found from this survey was that we need to have very clear guidelines on the variables to be uploaded, as well as very clear instructions for those uploading those variables. And that this process needs to be refined and streamlined to be able to have quality and harmonized data that can be used by many investigators. So the graph at the bottom of the screen shows what the different sites felt were barriers to submitting to NAC. The majority of people said that they were busy with other things, which makes sense. The ADRCs are busy, which really is key that we streamline things to make this a possible collection of data for everyone. Additionally, there was confusion on the process, so we need to make sure that we have very clear guidelines. So in looking at that original set of data and talking with the work group and working through all of the variables that we thought we would be interested in, we had additional questions that we wanted to ask the ADRC. So we created a new survey that was sent out in April 2024, so this year. We had 31 ADRCs respond, and we're very grateful to those ADRCs because we know it was a very quick turnaround, so we appreciate the time and effort that went into responding to us. So 29 out of 31 ADRCs were interested in providing biomarker data for blood or CSF to NAC. When asked how far back that data goes, I believe the furthest response we got was 2000. So there's a lot of data to be uploaded to NAC, which is fantastic. We found that there's a variety of in-house and external assay platforms utilized, which you can see in the bottom corner in that graph, outlines the different types of platforms utilized. And then we also found that there's a mix of corrected and uncorrected data. So some ADRCs only have corrected data, some only have uncorrected data, some have a mix of both, and some aren't sure yet because they're just starting to get their programs going. Additionally, to be able to formulate who would be the most likely person to be answering these or entering these variables, we asked each center that question, and we found that most centers consider having the data core or laboratory staff submit the data. So our major takeaway from these surveys is that the centers have a lot of data to share, which is fantastic, and we're very excited about this. But that the process of sharing that data really needs to be streamlined as well as clear to be able to make it as painless as possible on the centers. Additionally, the variables have to allow for many platforms, many data types, and many correction factors. And then the variables must capture enough information to make that data usable. So we want to make sure that the data can be combined for downstream analyses. So now I'm going to move into our recommended variables. So the work group met many times and worked through what we felt was important to be able to make that data usable by investigators. So in looking at this, I've broken it down into minimum requirements, desired information, and then information that if you have the time and willingness to provide, we would greatly appreciate. So within those groups, I've grouped the variables by site and specimen, lab and assay, and data and analysis related. So looking at the site and specimen variables, we would need the site that the data was collected at, the participant ID, the collection date of the sample, collection time, collection tube type, time to freeze, and then what type of specimen was analyzed. So was it plasma, CSF, et cetera. When we consider variables related to the lab and the assay, we would need to know what platform was run, what instrument model was utilized, who the manufacturer of the assay was, catalog number and lot number, as well as a lab name. So when we say lab name, we're talking about, was it an internal ADRC lab? Was it an external lab? Was it NICRAD? And that will allow us to utilize that information when we're searching through data that's available to look for labs that are the same. Additionally, we would want to know the instrument ID within the lab. If there's multiple instruments, we'd like to see whether there was variability between the two, the batch ID, and the date that the analysis was run. Now, when we're thinking about the data and analysis variables, we would need to know what biomarker was analyzed, so NFL, GFAP, A-betas. Additionally, what the concentration was of that biomarker. We recommend that it's a dilution-corrected concentration. Additionally, we would need to know the units, so picograms per mil, et cetera. We're asking were controls run, so that would be a yes or no answer for this one. And then, was the data corrected for lot, batch, and or instrument differences? Moving on to the desired variables. When we're looking at the site and specimen grouping, we would be interested in knowing if it was a fasting collection, how many freeze thaws occurred on that sample, the time to centrifugation, centrifugation speed, and centrifugation temperature. We'd also be interested in if there was a protocol deviation. So did something happen to this sample that didn't happen to the rest of your samples? And then for the data analysis question here, it's sort of a continuation of that first slide where we asked if there were controls, yes or no. This is asking what kind of controls were run. Was it a manufacturer-provided control? Was it an in-house pooled control? Perhaps it was both. And then finally, variables, if available, if your center has time and ability to give this information, that would be great. So fasting duration, needle type, gauge, storage tube type, position of the patient, and then temperature between that collection and centrifugation step. Additionally, I've also broken down some variables that would be for blood or CSF. So was a tourniquet used, or is there hemolysis in the blood? And then also for CSF, was blood collected at the same time as that CSF sample? And then were other routine assays run on that CSF? So next steps, first thing, we're gonna get your feedback. We're excited to present and hear your thoughts on this. And then we're going to work with the Biomarker Steering Committee to finalize these variables. And then following that, we will work with NAC to fine tune and implement the incorporation of these variables. And with that, I want to thank our work group members. I really appreciate everybody's willingness to participate and give of their time. I did a quick calculation and determined that we had about 14 different institutions involved in this. So we've had a lot of input from a lot of different groups. I need to thank our NIA representatives, so Cerise and Nina, as well as our NAC representatives who gave of their time and their effort to help set everything up and make sure they were able to answer our questions. And then special thank you to the Biomarker Core Steering Committee for all of their commitment and ability to answer our questions through this process. So I think we're gonna start taking questions.
Okay, so Kristen will stay up on the stage here. We have our other 3 representatives online, and we will take questions and feedback on what you heard this morning. So please go to the microphones. I think questions are optional online, which Hannah is going to monitor for us. So we'll start off with Dr. Seshadri.
Thank you. Thank you all for wonderful, very important presentation. I noticed that there was a question about not having vascular markers, and in the list, one of the things that didn't come up was O-link, and I wondered if it was not asked for because SomaLogic was.
Yes, so Tom or Tim, do you have an answer about whether O-link was included?
We were actually reporting back on what the centers told us, right? So it was not that O-link isn't being utilized or important, but it wasn't reported back from the centers survey.
Thank you. Yeah, Dr. Johnson.
Good morning. My question is about the existing ADC-FB protocol. I didn't hear a lot of mention of that. Will that existing pre-analytic blood collection protocol be sufficient for the future needs that you're planning, and how does that fit into your plans?
Yeah, so that's also a Tom and Tim question. I think you're working off of the existing ADC fluid biomarker best practice guidelines, is that right? Do you anticipate significant changes to that, I guess, would be the key to Sterling's question.
Yeah, so I guess that protocol is quite, sorry, just hearing a little bit of. That protocol is very good. I've looked at it myself. But one thing we would like to identify, are there multiple sites using the same protocol, or are there deviations? So if we get to the point of realizing that there are not many deviations from that standard protocol, then it might end up being what we propose is very similar to that one, right? But if there are also significant deviations, then we'll have to account for those and try to find a way of streamlining across sites.
Okay, thank you. Yeah, Dr. Glasgow.
Hi, so just a comment about the initiative to collect pre-existing data. It'll be a lot of work and a lot of moving parts. As a prelude, I would encourage the committee to take a look at the data that has been generated by Jeff Dage, which covers a bunch of different centers over the last approximately 3 or 4 years. Again, everybody would have used slightly different preparation methods and this, that, and the other, but I'm curious about how much variability there might be of reasonably standardized plasma biomarker data across centers, because a concern I have is as we go back in time, there may be a huge amount of inter-center variability in the big biomarker picture. I'd be just curious if you can get a handle on what the problem looks like in recent years when there has been an effort to try and do something centrally.
Yeah, so Kristen is the director of the NICRED Biomarker Lab. So Kristen, can you speak to that question?
Yes. So for ADCFB in particular, we have about 26 out of the entirety of the ADRCs utilizing the ADCFB protocol. So we have a relatively standardized protocol in place. I do know that some centers have some minor deviations to that. And as part of our effort to standardize things, we do track which centers do things a little bit differently. And so I think the key in this is knowing what the centers have done so that we can utilize information that exists already about the the outcome of that type of variability when we're looking to combine data sets.
So just to continue from that, have you done any analysis to see how that center-to-center variability affects the data that you're able to get in NICRED?
So there's a potential that Jeff may have already, but because we do have access to the ADCFB data, that is a very good possibility and probably a great place to start in answering that question in particular.
Okay, that's great. We'll go over here, please.
I didn't recognize everybody on the list, so is there a statistician in this group that will help with the data harmonization and looking at variability standardization?
Well, so for ADCFB, we do have people on our IU group that will help with that type of analysis for us. But I don't know if particularly, I think there might have been one person on the working group that had a was a statistician.
Yeah, I think there's one card-carrying statistician on your group. Dr. Jaiker.
So, I'd really like to applaud the group. This really is a notable effort, and it's incredibly complex. And really this to follow up on what Doug just said. But, even when we look at CLIA standardized rigor in testing, every individual clinical lab has to develop its own sets of normative ranges and values because there is inherent variability between labs. There always will be. So I think it's very important as you guys begin to tackle this. I didn't hear anything about looking at normative ranges across centers so that we can actually compare apples to oranges. I think this is a really important part because just generating data that has high variability or cannot be utilized center to center is problematic. I really see as we move forward, either we're going to have to embrace that or we are all going to have to send our samples to a single laboratory. This is what we do in clinical trials. And if you're doing local tests, you have to submit your normative value ranges for your center. If you're using a central lab for all samples, yes, they can be pooled because we know that we've reduced that variability. So could you address, are you guys tackling this issue? Because it really is the most critical issue, in my opinion.
Yeah, so maybe we'll go online first. Dr. Elahi, has your group discussed this at all?
Yeah, so this is really the beginning to understand factors that actually matter and to identify some centers that may be more similar to one another to start working towards that. I agree with the comment that was made. Ultimately, it may come down to the fact that we cannot really pool the data across the centers. There's really no clean way to do that, and that there's too much of a batch effect. And there are two different factors. There's the pre-freezing of the biospecimen and then the treatment of the specimen afterwards. And both of those have a lot of variability. So agreed, but we're not there yet. This was just the beginning.
Dr. Russ, you have something to add?
Sure. So I would say that going back is a little bit harder when you're looking at historical samples. And so that's why we really need to collect these variables so the investigator who wants to utilize it can make that call on whether or not they want to incorporate those historical samples. But I will give a plug for NICRED at this point because we do have a harmonization project going. So labs that are interested in looking at that variability and participating in a harmonization going forward would have the opportunity to do that as well.
I'm not sure if Tom or Tim, you have anything to add to this discussion before we move on?
Just a quick one to add that, yeah, I agree that this is really important, and that could be like the next step from this whole project that's being done. And then because even using a single center, still have like some issues, like you could have some variability between runs that still would need ascertaining. And especially now, if we're looking at different platforms, different instruments, and even different between their runs, that all could lead to some issues. That one of the ways that potentially we could go after this would be to maybe get some reference materials for the same assays, like assay targeting phosphatide 217 or 181, or whether it's mass spec or not. Then we could have like the same sort of like a peptide that we agree with, and that could be run at multiple sites. And based on that, we could have the statisticians have a go at trying to look at that data as again the quality control samples to try and work out, could we harmonize the data, because you would end up getting different values. But if you go in with this material, which is a reference method, could you be able to come out to like a central point? And that could become a resource that for every data that get generated could be compared to that. And maybe that could be a way going forward.
Thank you so much, Tom. We've got Dr. Maestro has been very patient, so I want to make sure she gets to ask her question before we move on to Dr. Schindler. So please go ahead.
Thank you very much. I'm just wondering how the diversity of the people actually are going to be considered when we look at the diversity or the variability of the results between the centers. I mean, comparing centers really account for all these techniques and methods and processes, but the people and the conditions in which they live might be a big difference here.
Because this is incorporated with the NAC data, I think that we will have all of those variables linked to the biomarkers as well.
Yeah. And I actually think, the beauty of being able to pull all of this data into NAC will give us much more diversity than we have center to center, so that we can address that exact question. Yeah. Thank you so much for bringing that up. Okay, so thank you, Kristen. And thank you to everyone, Tim, Tom, Fanny, thank you so much for joining us online, and we'll be taking some of these discussions back to the working groups. We're going to move on to Dr. Suzanne Schindler, who is a clinical dementia specialist and the fluid biomarker core leader at Washington University Knight ADRC. She's going to speak about the generalizability of fluid biomarkers. So thank you, Suzanne.
We're with you in person today, but it's my son's birthday, so I'm celebrating with him at home. I do hope you have a wonderful gathering in Austin. These primary disclosures previously analyzed biomarker data at Washington University. By Washington University through C2N Diagnostics and Roche Diagnostics. One of the themes of this meeting is generalizability. I'm going to discuss generalizability in the context of fluid biomarkers. We are increasingly using fluid biomarkers research, clinical trial, and clinical diagnosis. Although many of us about fluid biomarker context of research study, fluid biomarkers are increasingly being used in clinical care and may have a major impact on the lives of patients. Here is some CSF biomarker data from the Memory Clinic at Washington University, where we have scaled up biomarker testing over the last couple of years. Each of these points represents a patient with cognitive impairment who underwent CSF testing to determine whether amyloid pathology was present. Patients with a CSF p-tau 181 to A-beta 42 value above this red line are considered to be amyloid positive. And after integrating these biomarker results into a comprehensive evaluation, some of these patients were diagnosed with dementia due to Alzheimer's disease pathology. And additionally, based on these CSF biomarker results and many other factors, some of these patients were started on amyloid-lowering treatments. In contrast, patients below this red line are considered amyloid negative. Many of them were diagnosed with non-Alzheimer's disease causes of cognitive impairment, and they were not eligible for amyloid-lowering treatments. Because we are diagnosing and treating patients based on these fluid biomarker results, these tests need to be highly accurate. And they need to provide accurate results, not just in healthy, well-educated, non-Hispanic white individuals, but in everyone. So generalizability is important. How do we go about assessing the generalizability of a fluid biomarker test? I think an instructive way to think about this is with the concepts of signal, noise, and the signal-to-noise ratio. Biomarkers are useful when levels reflect key biological and/or clinical factors related to a disease of interest, like Alzheimer's disease. The association between biomarker levels and these disease factors is the signal. Biomarker levels may additionally be affected by factors not directly related to the disease of interest that partially obscure the signal. This is the noise. Generalizability refers to how consistently biomarkers reflect key biological and/or clinical factors across all individuals. You can think of this in terms of the signal-to-noise ratio. High generalizability requires a fluid biomarker to have a high signal-to-noise ratio to clearly see the underlying biological or clinical condition regardless of individual factors. It's like the right half of this photo. You can clearly see what's going on with each person. This is in contrast to fluid biomarkers with a low signal-to-noise ratio, which are like the left side of this photo. It's challenging to discern what's going on with each person, and it may be clearer in some people than in others. So for the rest of my talk, I'm going to discuss how to evaluate the signal, the noise, and the signal-to-noise ratio of fluid biomarker tests. Let's start by thinking about how to define a fluid biomarker signal. To assess this signal, you must have an excellent gold standard for key biological and/or clinical factors. A good example of this is amyloid and tau PET, which image AD pathology. High associations between fluid biomarkers and the reference standard demonstrate a strong signal and a high signal-to-noise ratio. In this study, we directly compared a high-accuracy blood test for percent p-tau217 to two FDA-approved CSF tests in classification of amyloid PET status, and found that the blood test and the CSF test performed very similarly with AUCs of 0.96 to 0.97. This kind of high performance tells us that plasma percent p-tau217 has a very strong signal in classifying amyloid PET status, at least in this particular cohort. While classification accuracies are highly relevant to clinical care, evaluating the continuous values of fluid biomarkers against reference standards is even more informative. We can assess the strength of the associations between continuous values for fluid biomarkers and reference standards. These kinds of analyses can reveal nonlinear relationships and different relationships during different phases of the disease. Some fluid biomarkers may reflect more than one pathology, and models can evaluate the association of fluid biomarkers with multiple pathologies simultaneously, such as with amyloid, shown here in blue, and tau, shown here in green. Longitudinal data sets may be particularly powerful in evaluating the signal. Biomarkers that initially appeared noisy may have a clearer signal once the longitudinal trajectories are examined. For example, once you adjust for differences in baseline levels, the trajectories for CSF percent p-tau 217 are quite consistent across individuals. Now let's talk about evaluating noise. With fluid biomarkers, there are many potential sources of noise. Here is a figure from a study led by Wagner Brum. The study collected plasma samples every week for 10 weeks from 20 healthy participants aged 40 to 60 years. When the same exact plasma sample was measured twice, there was a little noise, about 3 to 6% variance in biomarker levels. In the same individual, over 10 weeks, there was a little more noise in the biomarker levels, about 4 to 17% variance. Now, if you look across the entire group of healthy individuals, as you might expect, there is even more variance in the biomarkers, ranging from 6 to 30%, even though they were healthy controls. Given that some of the individuals in the study were a little bit older, up to age 60, it's possible that some of them had a mild degree of neurodegenerative pathology. So maybe there's a little bit of no real signal in here. Still, it seems that much of the variance shown here is related to factors that are unrelated to Alzheimer's disease. They're noise. Now, it's important to note that the changes associated with disease are in some cases several 100%, and not 20%. So while this level of noise isn't ideal, it still can be overcome by adequate signal. Still, this noise can partially obscure the signal, especially if the signal is low. A number of studies have demonstrated that fluid biomarker levels are associated with factors not directly related to the disease of interest. Biomarker levels may be affected by factors such as age, sex, race, ethnicity, comorbidities, even medications. These studies include seminal work by Michelle Mielke demonstrating associations with fluid biomarkers and medical conditions, including chronic kidney disease. Now, what I'm describing as noise in this context represents a heterogeneous set of variables, from social constructs like race to biological factors like EPOE genotype to factors like comorbidities that may be influenced by both social determinants of health and biological factors. This noise may be very interesting and informative. By calling it noise, I'm not meaning to diminish the importance, but rather just to say that these variables are factors that may affect fluid biomarker levels and thereby partially obscure the signal for AD pathology. Now, especially when lower performing biomarker tests are used in a diverse clinical population, such as was described recently in a paper by Boudeleau, the high noise may almost completely obscure the low signal to the point that the biomarker levels have little relationship to the disease. Instead, the biomarker levels better reflect non-disease related factors. This is an important lesson. Lower performing biomarker tests in a real world population have poor generalizability and therefore relatively little utility in many contexts. Now, one issue that has been particularly concerning to the field is differences in biomarkers by race. A number of ADRCs, including WashU, UPenn, Wisconsin, and the Alabama ADRCs, as well as the Harvard Aging Brain Study, contributed to this study led by Kenji Zhang that found different relationships between CSF biomarkers and cognitive measures in Black and white individuals. Additionally, a growing number of studies have found differences in the rates of amyloid positivity by race and ethnicity, even after accounting for many other variables. It's unclear if this is just an effect of different recruitment methodologies or whether other factors are at work here. However, as demonstrated in a very recent paper by Doris Molina Henry, lower rates of amyloid positivity in Black and Hispanic individuals recruited for clinical trials may exacerbate the underrepresentation of some minoritized groups in AD clinical trials. Now, classification accuracy is a clinically relevant way to consider noise in fluid biomarkers because clinicians typically look at the positive or negative result. At a certain biomarker value, do all individuals have the same likelihood of being positive? Does the value mean the same thing in every person? Or do some factors make it more or less likely that one individual is biomarker positive, even if they have the same exact biomarker value as another person? In this study that I did with Tommy Kakari and others, that included Black and white individuals matched by key covariates, we found a high-performance plasma A-beta 42-40 test accurately classified amyloid status regardless of racial group. However, Black individuals were less likely than white individuals to be amyloid positive at the same exact levels of p-tau 181, p-tau 231, and NFL. Now let's think about these concepts of signal and noise using these models. In logistic regression models of amyloid status, covariate adjusted biomarker effects reflect the signal, covariate effects may reflect the noise, and the AUC reflects the signal to noise ratio. For biomarker tests with a high signal to noise ratio, such as plasma A-beta 42-40 in this study, biomarkers have a strong association with amyloid status. And covariates, such as race, have weaker associations with amyloid status. Now, for biomarkers tests with lower signal-to-noise ratio and lower AUCs, such as plasma p-tau 181 in the study, biomarkers have weaker associations with amyloid status. And then covariates, such as race, have stronger associations with amyloid status. Now, what this suggests is that high accuracy tests, where there is a strong association between fluid biomarker levels and underlying pathology, may mitigate some of the effects of covariates. Overall, we need to be using high accuracy tests for good generalizability. Here's an example of a high accuracy blood test, the PrecivityAD2 test that combines plasma A-beta 42-40 and % p-tau 217 into a modeled value called the amyloid probability score 2, or APS2. In our night ADRC cohort, this test classifies amyloid status as determined by CSF or amyloid PET with an AUC of 0.96, and there is no significant effect of race, sex, age, hypertension, diabetes, or BMI. Now, unfortunately, we did not have data on kidney function, but previous work in the BioFinder cohort has found that this % p-tau 217 measure was less affected by kidney function than p-tau 217 concentration. The use of biomarker ratios generally may be helpful in mitigating the effects of some factors on biomarker levels. Now, I would note that even a perfect blood test would not get to an AUC of 1.0 because the reference standards we have of CSF tests and amyloid PET have some degree of noise by themselves. And the best we can probably ever achieve in this particular cohort is about 0.97. Overall, what this data suggests is that tests with a high signal-to-noise ratio may be generalizable. Now, of course, we need to evaluate this specific test in an even larger and more diverse cohort. We need more representative cohorts, and many of us are working on developing them. Research cohorts traditionally have been relatively homogeneous, so you would expect them to have lower biomarker variance than the general population. This means that biomarker tests are likely to be less accurate in the general population than in research cohorts. Ideally, cohorts used for biomarker validation would resemble the intended use population in terms of key covariates. For example, if you're developing a screening test, it would make sense to validate this test in a community-based cohort that resembles the population you want to screen. If you're developing a diagnostic test, it would make sense to validate this test in a memory clinic cohort that resembles the population you expect to diagnose. However, one obstacle is that biomarker validation requires high-quality reference standards, like CSF tests and PET, and typically requires patients to consent to be in research studies. Many individuals in the general population do not want to be in a research study, or they don't want to undergo CSF testing or PET. So, it may be difficult or even impossible to obtain CSF and PET in a truly representative cohort. One solution is smaller, targeted, and carefully designed studies. For example, if you want to test whether or study whether a test is affected by chronic kidney disease, you can enroll a relatively small matched cohort of individuals with and without chronic kidney disease and answer this question relatively well. This approach may be more efficient than studying a large population with a low rate of kidney disease. In conclusion, we need fluid biomarkers that accurately and consistently reflect disease processes in all individuals, and high generalizability requires a high signal-to-noise ratio to clearly see the underlying biological or clinical condition. There are a variety of study types that can be used to assess the signal-to-noise ratio of fluid biomarker tests. We need more representative cohorts with reference standards to validate fluid biomarkers. However, smaller, carefully designed cohorts may also be helpful in answering specific questions. And finally, I would say that I'm optimistic that we will be able to develop and validate high-performance fluid biomarker tests, including blood tests, that will enable more accurate diagnosis of AD pathology in all individuals. I would like to acknowledge our research participants and their supportive families who have enabled all the work that I described. I also thank my mentors, collaborators, clinician colleagues at WashU and at BioFinder. Thank you very much.
Thank you so much, Dr. Schindler, for that great talk. Our next presenter is Dr. Alison Krause of Cleveland Clinic. And Dr. Krause is an assistant professor of pathology at Case Western Reserve and co-investigator with the neuropath and biomarker cores of the Cleveland ADRC. So I apologize, I said you were from Cleveland Clinic, you're from Case Western. She's going to speak about biomarkers of alpha-synuclein pathologies. Thank you.
Thank you.
Okay, so I think we still have Dr. Schindler online, I hope, for questions. And obviously, Dr. Krause is here, so if you we'll start with questions for Dr. Krause and Dr. Schindler, and then if you have any other discussion points and we have time, Dr. Russ is also here. So yeah, Dr. Rabinovich.
Thanks, Donna. For Alison, very nice talk. Two quick questions. One is, you showed the 4R seed amplification assay. I haven't heard a lot about that. How validated is it? How close is it for potential diagnostic use?
Yeah, so that's an example as it was applied to brain tissue analysis. There's some pilot studies indicating some utility in CSF, but that is still development. For alpha-synuclein certainly much farther along in the biofluid realm than any tau-based assay would be.
And then just another question. Could you talk a little bit about the feasibility of using these seed amplification assays from blood samples as opposed to CSF?
Yeah, so the value in it is undeniable. There are reports coming out that these can be applied in these settings, and I think requires larger validation and certainly some methodological comparisons of how reproducible, how sensitive this is, and limitations that might be had.
But it should be feasible, you think?
Should be feasible.
Okay.
Okay, Dr. Sano.
Sure. Thank you. Thank you. All fabulous presentations. I actually wanted to comment from Dr. Schindler's talk, where she makes the observation of how important it is for us to continue to collect the CSF or the amyloid plus the blood biomarkers. And it raised for me an issue where many cohorts are actually being collected with the more expensive or burdensome procedures only based on positivity of a biomarker. And I think it might be important as we collect data to actually be very careful of what the protocol was for collecting that data, because it can flood variables or cases that create a self-fulfilling prophecy. So I think it's really important to consider criteria that we use to collect the cohort when we try to put these data sets together.
Yes, I certainly agree that we have to be very careful on how we recruit and enroll and keep participants in studies. The way the cohort is set up can really affect the conclusions we reach.
Thanks, Suzanne. So we have an online question, then we'll come over here. Hannah?
Yes, hi. We have a question from Warren Barker. It's actually for Dr. Russ and the biomarker working groups. Can you address the cost of plasma biomarker analysis for historical samples and prospective samples?
So the cost, like the specific costs? Well, I mean, the cost would technically be the same. So if you were, I only have the perspective of NICRAD, but so if you were to submit your historical or your current plasma to us, then we have, certain prices that depend on which assays that you're interested in. However, if you're part of the ADCFB initiative through your ADRC, then you provide NICRAD samples, a certain volume, and then you receive those biomarker analyses for free as part of that initiative. Also, I'm sure my contact information is available, so feel free to email if you have a more specific question.
Thanks, Kristen. Yeah, we'll go to this mic. Thanks.
Great presentation. My name is Dr. Pinar Koskun from University of Kentucky. So I appreciate, all of us appreciate the variability of the biomarkers. And I was actually pondering the idea, would you consider actually, or would it be a good idea to do continuous, like the generating curves or trends from the patients who are a subjective memory problem, and then do curves and then compare those to overcome this conundrum wrapped into dilemma, actually. So I was wondering, that could be a solution, because intrapersonal differences also very important, and maybe the disease progress is that we just only capture one point of care value, so that curves may be much more valuable, that I was thinking.
Yes, so I agree that longitudinal studies are extremely, and in fact, we see that when we look at longitudinal curves, we often see a lot less variability than when we look at cross-sectional analyses, because you're using each person as a control for themselves. So I agree that that's very important to do. Of course, it takes a lot of time and money to get that kind of data. So that's just one downside to that. So it's very helpful, but it does take quite a bit of time and money to get a lot of longitudinal data on many individuals.
Thank you.
Okay, do we have any more questions for the group? If not, we will thank all of the speakers for their great presentations, and thank you all for coming.